presentation shall commence momentarily. Kindly review forward-looking statements.
The majority of the work ongoing right now in the lab is really related according to photodynamic therapy. So again, for those who don't know, the use of light-activated drugs in oncology and also some other indications. This complicated picture here on the side is really only what we think is actually ongoing during photodynamic therapy. That is, our drug is absorbing light, it's undergoing some physical processes within the molecule itself. So those physical processes allows it then to react with molecular oxygen that is inside the tissue. And through those reactions, we are creating what we call reactive oxygen species, that is singlet oxygen predominantly and the radicals. Why do we like it? Well, when we inject the drug or administer the drug, it's non-toxic. So the side effects to the patient are completely minimized. The cytotoxic substances, the reactive oxygen species, are extremely short-lived. We are talking about typical microseconds. So they cannot go very far from the point where they are produced. They typically stay within the cell where they are produced, killing off that particular cell. The photosensitizer itself is what is commonly known as a catalyst. So it can basically, a single of those molecules can produce 1,000 of those cytotoxic molecules. That overall is again limiting the actually amount of drug we have to administer to the patient at that time. This one down here in the structure is TLD 1433. The at this point most promising ruthenium compound that was evaluated and tested in the lab, and that is also the compound which is going right now into clinical trials, and my colleagues, medical colleagues, will then basically later present on this. In the bladder cancer, we use the compound as is. For future systemic administration for other tumors, and we're looking right now basically at brain and some other tumor models at this point preclinically. We have found out that, again, together with the company, with Theralase, that the compound binds very strongly to what is called transferrin. Transferrin you have in your body as an iron transporter. You need iron to make heme, for example, or hemoglobin. And through the binding with transferrin, we basically can exploit what is called transferrin receptors, which are on all cell surfaces. The big difference is that a normal cell has a much lower expression of those transferrin receptors compared to a tumor cell. Cancer cells really need a lot of iron. They have this receptor overexpressed. And that basically will allow us to have a nice differential uptake in tumor cells versus in normal cells. After the uptake, we basically can then irradiate those cells and again, ultimately will destroy the cells. There's a couple of other very interesting side effects to that transferrin story. It makes our drug better soluble, and it also reduces even the toxicity of that drug even further. While we generally need to administer only a fraction of the dose where our drug previously showed toxicity, that differential between when the drug itself becomes toxic versus with the transferrin is much, much larger at this point. So it's again increased the actual profile or the safety profile of the drug itself. And that was basically also demonstrated in what is called GLPs, or good laboratory practices, in pharmacologic and the toxicology studies, which were executed over the last year. So the drug story on the one side is very well developed, and Theralase, under the guidance from Arkady Mandel, has essentially the drug manufacturing, drug stability, quality assurance on the drug side excellently under control. Where we are coming back in is, again, to provide a much better dosimetry. Some of you may know that photodynamic therapy for bladder cancer was the very first indication approved for photodynamic therapy, and it was approved here in Canada in 1993. But the original drug that was used was systemically injected, did not have a very good selectivity, and then there was red light that was used to activate the drug. And with what happened with the activation of that particular drug that had a lot of other disadvantages, what happens is that not only the tumor was sensitized to the light treatment, but also the muscle layers which help the bladder to contract and expand. And there was a delayed death in some of those muscle cells. That layer became rather rigid. It could no longer expand and contract, and that resulted ultimately in incontinence to the patients. And the rate of incontinence that developed was too high, and it really fell out of favor for the urologists. Some of the problems were already back then identified, but the other drug companies that actually was driving the process were not listening to some of the physicists who basically stated you need to have very good dosimetry. The surface of a healthy bladder is, if you would look through it through a cystoscope, appears white. So there's a lot of light that is backscattered. And this room, if you would have painted the colors of the wall, not in Theralase green, but in white, we would actually have more light in here, because the light from the lamps would be reflected from the wall, and from there the white would reflect it coming back in here. If we would have painted it darker color or black, it would be much darker in here, obviously, having the same amount of light. And you can think about the bladder itself being exactly the same. Yeah? A healthy bladder has a lot of light reflected, but the more and more of disease you have in your bladder, the darker it gets. And that really affected how much light ultimately was inside the bladder and how much light could activate the bladder. So there was a lot of problems with over or under treatment. Theoretical studies basically showed that can be easily up to a factor of 4 or 5. Yeah. So we have basically immediately said very early on, we have to combine the drug itself with a very controlled light delivery and also light dosimetry system to really concentrate a lot better the dose. Yeah? Your physician, he's going to tell you, if you have a particular pain, take 500 milligrams of aspirin. More or less the same, independent of your body weight. He's not telling you, oh, take anywhere between 50 and 5,000 milligrams of aspirin every day. Yeah? That range would be just too large, and that's what we have essentially to combat, and that's what we really wanted to control. So we basically built a dosimetry system here with the engineering staff that was the experienced engineering staff at Theralase, to ultimately provide the treating physician then with a direct feedback of how many photons have we actually right now delivered per surface area of the bladder surface. We want to treat the entire surface area, but we have to control it. So again, the system we have designed and the ultimate probes with the consumable we have designed right now is providing that feedback. So the drug is basically instilled directly into the bladder. It's going, then after about an hour, it's going to be washed out 3 times just through the cystoscope directly. And ultimately, when we are then ready, we're going to insert our probe. We'll show what that looks like in a second, and then inflate the bladder to a previously predetermined volume. I will show you then later how we actually go through the whole process, how the physicians have now some feedback to make sure that the bladder, independent of its shape, can reasonably homogeneously be emitted. We're also using green light. Green light is much stronger absorbed in tissue than red light. And that gives me, again, a very steep gradient or difference in the dose I'm delivering to the bladder wall. Very high dose on the luminal surface, that's where our tumors are. The dose that actually gets to the bladder itself is significant, to the muscle is significantly reduced, And we have, even in much thinner bladders of some animals, we have not seen any damage to the muscle layer. And then the physician basically sees on the screens when we have actually reached our target dose, which is 90 joules per square centimeter. It's going to take about 45 minutes, an hour, depending on bladder size. And then we can basically stop the delivery. The hardware, that's what it looks like right now. Have all the safety stickers we need, pass by the biomedical department at the hospital. On this side, essentially, you have the laser. On this side, you have the dosimetry device. It was all built in-house. We have basically two systems, computers which are controlling the system, one for the laser and one for the dosimetry devices. In the OR, what we basically have, we connect the consumable here, which is held here in the hand. That's not in the OR, that's in our labs, obviously. To the laser itself, we connect the detector, simple connectors. We have here basically a device that allows us to measure the absolute power we are delivering at any given time. When we light it up, it has this nice green glow, and we can now verify here how much power is our fiber optic actually delivering at this point. We're actually going up to about 3 watts of green light. Doesn't sound a lot, yeah, but it's a very bright light, single wavelengths. You probably would not like to look at it once the system is hooked up and is actually working along. This is basically then the cystoscope that the urologist has itself. We basically use that cystoscope ultimately to insert our assembly, which is comprised out of a light delivery point emitter and basically those 3 arms, and each of those arms holds a total of 4 sensors. So at a total of 12 positions, we actually have a direct feedback of the energy or the power density, or then integrated over time, the energy density we are delivering. Why is that important? Again, as a physicist, we would think the bladder is round and spherical. It's not. All the other organs which you have in your peritoneum are nicely pressing around it. And a bladder can be nice and oblique like this. And if you have basically absolutely no feedback, you see that if you have a single point and we put it in there, we have a higher dose here, on here, lower dose on this side. Through this treatment, the physician has, and the urologist has, essentially the chance to do a slight repositioning of the source if you notice there's some overtreatment, some areas being treated too fast, some areas going to be treated too light. This is, for example, the bladder of a male, where the prostate, which would have been sitting down here, really deformed it very significantly. Again, the color code gives you the differences in the surface power density we would be delivering. And that basically allows us that we really eliminate any type of over-treatment or under-treatment, that we're getting as much of the surface area really to the target dose as we are desiring it. The device basically gives multiple tools here to the physician. We have here a screen where we basically can look at the fiber positioning. These are our 12 probes. And we can basically see that they have roughly the same power. This is now power density. Once we are happy with this, we can basically switch streams to what is called the dosimetry screen, where we have any type of our target we want to hear. We even tell the physician how long it probably will take until we have delivered all of the total power to it. Then we basically have to irradiate it, yeah? As I mentioned earlier, will take, depending on the bladder size, somewhere around 40-45 minutes, can be up to an hour. We are not allowed to go over an hour because physicians and surgeons are really impatient people. They want to get going. So we have a couple of restrictions, that we have to have a device that is actually satisfying the need of the physician and actually of the entire organization at that point. We start treating once we actually have reached our target. That is essentially when then the physician says, okay, stop light delivery, we turn the system off, the catheter can be taken out, and then we do all the other procedures the patient needs to do, we have to do during that phase 1 clinical trial. So the hardware is completely ready right now. It's been tested, and yeah, right now we're basically only looking forward to seeing when we get the first patient, going through the screening period of those patients, and then we want to treat it. And at that point, without further ado, I pass it over.
So my name is Dr. Girish Kulkarni. I'm a urologic oncologist at UHN and Princess Margaret Hospital. My specialization is in bladder cancer, and for the most part, I'm running the clinical trials for bladder cancer at this institution. So one of these clinical trials is the Theralase TLD 1433 Phase 1B. So to speak to what Dr. Jewett said, this is a safety trial. The main idea is to ensure that this drug and approach is safe in patients, and once we prove that this is the case, then Phase 2 and 3 are efficacy. So I thought the best way to start is a primer on what bladder cancer is. I know Dr. Lilge talked mainly about the science behind photodynamic therapy and showed some of the equipment. But bladder cancer is an unregulated growth of cells that are of the inner lining of the bladder, so where the urine is in contact. Those cells are called urothelial cells, so the cancer is called urothelial carcinoma. It's the 5th most common cancer, actually. It just doesn't get as much play as, say, prostate, breast, colon, but it's right there, right underneath. So this is a schema or a cartoon of a bladder. So a normal bladder would be a layer like this, coming down. And this is the urothelial layer. This is the lamina propria. This is important for staging. This is how we speak to each other and categorize tumors. This is the muscle of the bladder, so that's what gives it the force to urinate, and this is the fat around the bladder. Tumors that are confined to the urothelium layer are called carcinoma in situ, or if they're just growing on top, TA tumors. Tumors that have started to show invasion into the next layer down, that's the lamina propria layer, or T1s. And then T2s and T3s are the bad actors. So that's not what we're talking about today. We're talking about these 3 here, carcinoma in situ, TA, and T1. Those are 75 to 80% of bladder cancers. The ones on the far right are the bad actors. So we're trying to prevent some of these from turning into those. Anything that's T2 and beyond is called muscle invasive, going in the muscle. So these are non-muscle invasive tumors. The way we diagnose and treat them is with a scraping procedure. Here you can see a cartoon of that, where we put a scope through the urethra and scrape the tumor out. Problem is you can't necessarily say that you have all of it, and or the tumors have a tendency to recur in other parts of the bladder. So that's why we'll often give the high grade, the worst actors of the non-muscle invasive tumors, additional treatments in the bladder. So BCG is the most commonly used treatment. It's immunotherapy. Mitomycin C is chemotherapy. So what is BCG? It's live attenuated Mycobacterium bovis. So that's cow TB. It's still alive, but it doesn't have the same virulence or the ability to cause infection, but it can elicit an immune response. So the appeal here is that we're inciting the patient's own immune system to fight cancer. And that's actually very hot right now in terms of cancer therapies. Even though this was discovered in Queens, so it's Canadian discovery used around the world, and it's been around for 30 plus years. The exact mechanism isn't known. There's a number of steps of how BCG works, but that still requires some research. But we do know it's the most effective agent for high-grade tumors. It helps prevent recurrence, it helps prevent progressions, and that means advancement to the higher stage, the muscle-invasive categories. So how is BCG administered? This is important just because when we're talking about who's going to go into this trial, they have to fail a number of BCGs. So I just want to illustrate this point. There's a famous criteria or a protocol for how BCG is administered. Overall, patients get 27 installations over 3 years. Each installation is a week. So if you have a diagnosis of a high-grade tumor, the patient will then get 6 weeks. Then we look inside. If everything's clear, they get three more weeks. If everything's cleared, we get a 6-month look inside. Then they get 3 more, then 9 and 12 months look inside, then 3 more, and this goes on for another 2 years. So there's a lot of treatment burden for these patients. And say at this point, a tumor recurs, you start all over again. So these patients are experiencing a lot of treatments, and these tumors tend to recur, 40, 50, 80% recurrence rates. So what happens if BCG fails, as it does in about 30 or 40% of the time? You can re-challenge with more BCG, as I said, but ultimately, if the definition of insanity is doing the same thing over and over and expecting a different outcome, right? So if you keep giving BCG and it's not working, it's not going to work. And all these other agents, these chemos, mitomycin, gemcitabine, they don't really work that well either. At best, you're going to get 10, 20% responses, maybe 30% at 2 years. So ultimately, people will fail. And then the next treatment is remove the bladder. And if the bladder is removed, here's the kidneys, they make the urine. There's no bladder for them to go to, so we create stomas. And it's a big operation. It's disfiguring. Patients, for obvious reasons, don't like it. But if it's that versus die of bladder cancer, you're going to take it. And we can sometimes create new bladders at a valve for them. But the goal is to try to prevent as many patients from reaching this stage. So that's where this photosensitizer comes in, TLD 1433. It preferentially binds to, or at least one of the theories is it'll bind to this protein transferrin. And that's preferentially taken up by tumor cells as opposed to normal cells. And then when exposed to the green light that Lothar was talking about, reactive oxygen species are released. That's the theory, and it kills the tumor cell. Preclinical studies in rodent models demonstrate excellent efficacy, but we have to prove that in humans before we can actually adopt it. So that's where this trial comes in. It's a phase 1B trial, so that just means we're starting off. It's a safety trial, and patients need to have high-grade, non-muscle invasive bladder cancer of these stages that I talked about before. And the reason I went through that whole BCG regimen was just to state that they have to be intolerant patients, meaning they can't tolerate it. It causes a lot of symptoms. They say, forget it, I can't tolerate this. I got blood in the urine, I'm getting infections, it's burning. Or they have to have failed, meaning the tumors recur. So there's strict criteria for what constitutes a failure. So they have to get 5 of those first 6 that I mentioned, and 2 of the next 3, and then fail. Or if, like I said, if you fail early on or at any time point, we restart the clock. We'd give another 6. So they could have 5 plus 5 or 5 plus 2. So that's basically, they have to have had some exposure to BCG to be considered a failure, up to a year out of finishing BCG even. So that's been debated, and how we define BCG unresponsive in the bladder cancer world for a while, and all the clinical trials in the States that are running are using definitions like this, and this is what the FDA wants. So whatever the FDA wants, usually Health Canada wants the same. So the study objectives are, the primary one is evaluate safety. We need to prove that it's safe so we can go to the next level. So we're going to, as Dr. Jewett mentioned, start with a lower dose and go to a higher dose and measure adverse events and pharmacokinetics of the drug in the blood and the urine. And that's the next, the secondary outcome, the pharmacokinetics. So what concentrations are there in the blood and the urine, and the maximum concentrations. We're going to have the data on how well it works in these 9 patients anyway, so we're going to use that as well. So that's an exploratory or a second or tertiary outcome for those patients. But you can't make conclusions or definitive conclusions on 9 patients, but nevertheless, we'll get an idea as well. So this is the study design. So I'll walk you through this. If a patient is identified, there's a 2-week, they have their scraping. They've already failed a number of BCGs. We do a scraping because we saw something in the bladder and it comes back as a high-grade tumor. We've cleared them of their tumor temporarily, but we know there's a high chance this is going to come back. So when they come to talk to us at that point, we have a 2-week period approximately to screen them and say, okay, yeah, they failed the appropriate number of BCGs and they're interested in the trial. When we establish that, we obtain consent from the patient, and then we need to bring them to the operating room again. And at that point, we instill 0.35 milligrams per centimeter squared, expose them to light, the bladder to light, activate the drug. So we activate the photosensitizer, and then we follow them afterwards. So we have visits at day 2, day 7, then 1 month, 2 months, 3 months, and 6 months, and then we stop. So it's a 6-month safety period. We're starting with the lower dose, and then after we have the first 3 subjects, we're going to have our data safety monitoring board review the results. If there's a significant safety signal and patients are doing very poorly, then we have to stop the study. If there's a safety signal that's not significant, what's significant is that's hard to know. The Data Safety Monitoring Committee will probably tell us. Death obviously is significant, but we don't anticipate that. Then we'll continue on, enroll 3 more. If there is no safety signal, patients fly through, they're doing well, then we'll go to part 2, which is increasing the dose, doubling the dose, and repeating everything. So the idea is to try to do the first 3, prove that there isn't a safety issue, and then do the next 6. We don't know, right? That's the whole purpose of doing the trial. But it worked well in all the preclinical studies, so that's why we're at this stage. And that's why our research ethics board and our institution and everyone has approved it, and I'll get to that point. So this is the 2 weeks beforehand. This is important. So there's a voiding diary. So how much do we put into the patient's bladder? So they come in, getting ready for their laser treatment. For 1 hour before they are to undergo the procedure, they have to hold the agent, the photosensitizer, in their bladder. But everyone's bladder has a different capacity, and individuals may have different reactions to the environment. They may be nervous, etc. So what we do is we do a voiding diary, and that's 3 days of having a patient record how much they're urinating. This is common in urology. It's a very simple tool. You urinate into some measurement device. It can be a beaker. We usually give them a device that just measures the number of ml, and we get an idea for each void how much they're voiding, and we can get an average of what their bladder capacity is, and it's relatively accurate actually. Then we're going to take 80% of that, because we need to figure out, their patients are supposed to hold the drug for an hour. So if you give them 100% of their capacity and tell them to hold it, they're still making pee. So then they're at 120. We don't want this to be an hour of torture before they go in. They need to be relatively comfortable. So we'll go 80%. They're not to drink anything the night before. So that will decrease the amount that they're filling their bladder as well. And then we have a set amount for a certain voided volume or bladder capacity. We know what their surface area is and how much drug to give them. So that's all defined in the protocol. I think Lothar went over this, but basically we'll fluid restrict, like I said, because we don't want them filling the bladders quickly in the preoperative area. We put the catheter in, the bladder is drained, so they're empty, then we put the drug in, they hold it for an hour. In that time frame, the tumor, or whatever tumor is left, will be preferentially taking up the drug. Then we bring them to the operating room, and I think this is where I walked in. So I apologize if there's a little bit of overlap with what Dr. Lilge spoke about, but this is the only, I think, overlap slide. The bladder is washed 3 times to get rid of any existing drug in the bladder, so that's preferentially only in the tumor cells. We insert the optical fiber. It's through one of our special scopes. I have a picture of that as well. And we're going to have 1 hour of green light illumination because we're impatient. We can only do 1 hour. Okay? So where are we now? I was here last year, and we've made a lot of progress. We're more or less open. Okay? So the protocol is finalized. Our research ethics has been approved at the institution. The institution has given us authorization. Health Canada has approved the agent and the trial. All of the pharmacy, pathology, all the departments are on board. We've even had a medical advisory meeting with, these are world experts actually in bladder cancer, so that we can get independent input on the protocol. And that was in San Diego last year at one of our own national meetings.
After you do the green light for an hour, what happens?
Oh, okay. So after the green light's done for an hour, the light is turned off, the scope is removed, a catheter will be placed within the bladder because we need to continue to drain the bladder. The patient will wake up, and the aim is for this to be an outpatient procedure. So they'll be monitored in the outpatient area, the post-op area. And then I don't have all the other details, but the pharmacokinetic studies then occur at that point. So there's blood draws and urine samples at 1 hour, 8 hours, 72 hours to measure the amount of drug in the blood and the urine. So patients will go home the same day. They'll likely go home either after the 8-hour draw or they'll come back for that.
The company says that when the cancer uptakes the compound, it fluoresces. Is there anything in the process that would go in and look for fluorescing cells after the process?
It doesn't fluoresce. It takes up the cells and it's activated by the light.
Yeah.
So we're not actually looking for a fluorescent signal. I don't anticipate us seeing anything like that because we're going to be looking, we have a scope in the bladder for an hour, but we're not looking around at tumor sites, for example, or where we think the tumor is. We're trying to keep the scope centered and the laser fiber centered so that it's emitting a uniform light throughout the bladder.
Sure.
But it's not like, you may hear of some of fluorescent types of surgery where you can use a fluorescent dye to identify tumor cells and then you know to remove them. That's not the purpose here. Here it's preferential uptake of drug into tumor cells and activate. So theoretically, regular cancer, non-cancer cells of the lining of the bladder could take it up and they get activated. That's where the adverse events could come in.
Sure.
Right?
Well, actually, I was thinking that more of proof that you've completed the process, that all the cancer's gone after the fact.
Well, that'll come when we continue on at 1 month, 2 months, 3 months, when we start doing the cystoscopies to assess whether there's recurrence. Yeah. So the way we'll know for sure is if something returns. But we don't know at that time if every cell is killed. Just like with BCG, we have no idea. We just know that, and just like when if a patient gets chemotherapy for advanced breast cancer, we don't know. We just know that if we give it versus if we don't, there's a 20 or 30 or 40% improvement in survival. And the hard thing is actually trying to figure out who is the beneficiary, and that's what the concept of personalized or precision medicine is. We're trying to work on that in different avenues of cancer. I'm almost done, and then we can do more questions. So this is a dry run. We did in the OR as well. Okay, so we want to make sure we worked out all the kinks, and this happened about a month or so ago. So this is the basic setup of how the patient is going to be on the table. They're in stirrups. I've blacked out the face, so here's the device. So it'll be advanced a bit more than this, obviously it's in the bladder, but we have a specialized holder for the scope because it's supposed to be held in place for an hour, and I don't want to do that for an hour. I need to get coffee or something while this is going on. No, I'll be there, but this actually prevents motion as well. And so that will be within the bladder, and here you can see the light from the scope, and there's going to be a green light emitted as well. So our study is open. We're actively screening patients now. We completed a site initiation visit as well. So the appeals to patients of this is obvious. The number one is obvious. They get to keep their bladder, but no one wants to live with an ostomy. The operation to get to that point, even if you have a new bladder made, has a mortality rate of approximately 5% in the province. So it's not a small undertaking, and about 2/3 of patients have complications afterwards. So whatever we can do to actually save bladders will be beneficial to our patients. So that's what the avoid cystectomy line is. The nice thing about this is, so we have other trials as well, but the benefit of this trial compared to other ones that are around are a lot of the other trials, patients have to keep coming back. Remember I showed you the BCG regimen of 27 BCGs and scopes all the time. We still have to scope a lot here, right? Because we have to, that's the way we determine if there's recurrence, but it's a one and done. So that's the beauty of this approach. So it's good for non-Toronto patients. If you have to come twice a week for 12 weeks and then every week for 2 years, you can't do that if you live in Sault Ste. Marie, unless you're rich. So they can come down, get their treatment, and they still have to come for the protocol follow-up, but that's much more manageable than what some other agents that are being brought to market. So that's a great appeal for a country like Canada. And it allows us to be part of something new and innovative, especially for the patients. So I do have a patient who I've spoken to already, and he's identified and ready for inclusion. He held off on his operation. We were going to do it in December because he wants to do it after the holidays. So the first one we should have in January. So why we think we're the right center for this, we're in the top 3 in terms of research for cancer in the world. We're a very high volume bladder practice. We have a specialized multidisciplinary bladder cancer clinic, the only one in Canada, and one of probably the top 2 or 3 in North America. And we also have a collaborative with other urologists around town who are aware of our studies, and they're interested in sending patients to us because the patients are given the choice of PMH clinical trial or remove your bladder, and many are willing to undergo the trial. So I've told you who I am. So I'm on the Bladder Cancer Canada Advisory Board. I have a number of positions now at CCO in Cancer Care Ontario and bladder cancer that I haven't put there. You've met Lothar. Our research coordinators are not here, Mike Nesbitt and Kathy Lee. And we have a number of co-investigators, and Mike Jewett has been involved since the inception, but these are our other colleagues who can also refer patients. And now I guess we're time for questions.
The theory is that the compound attaches to or binds with the transferrin. How would that happen in the bladder? I know Lothar said that there could be residual in the urine, but it's a curious question. If that's the mediator to get through the cell membrane, is the transferrin. How the compound can't do it alone.
No, so maybe Lothar can address this, yeah.
Let me answer that question. The compound by itself can get into the cells. The process is just a lot more effective if we have it with the transfer. Right now, in the bladder, if we inject it, we have shown in preclinical studies that essentially we have an effective treatment depth of easily up to 1.5 millimeters. So the compound goes into the cells, into the tumor cells, and it diffuses through the tumor cells itself. Yeah. And so that would be one of the reasons. There is always some transferrin in the urine, in the vessel. All those patients have a certain amount of bleeding. Yeah. So it's probably both of those effects at the same time. But for the bladder cancer trial, we really do not need the transferrin at this point. Yeah.
And remember, these patients have had their bladders visibly cleared of all tumor. So that it's not, we in urology, we have marker lesion studies where you just leave a tumor and give a drug and see does it get rid of that lesion. That's not the purpose here. The goal here is it's almost as an adjuvant treatment. So in addition to the scraping out of all the tumor, because we know that the tumors recur, so there must be microscopic amounts left. So it's to treat the microscopic amounts that we can't macroscopically remove.
Having said that, do you see the protocol changing over time with this compound? Rather than do the scraping, just go straight to the compound?
Those are tougher to do, the marker study lesions, because of like to get ethical approval to leave a tumor in someone is difficult to do. I don't anticipate that people wanting to do that.
No, I was thinking more as a primary protocol. You find that there's a tumor and you treat it with a compound.
As opposed to scraping it? Yeah.
After efficacy is demonstrated, you mean?
Yes.
So a new market, you might say.
Well, yeah, the goal is, if I'm interpreting correctly, is this would hopefully be the treatment for BCG unresponsive patients. And then another question could be, could we use it instead of BCG? That would be another trial, right? So we could.
Surgery.
Possible. I figure that's what you're getting at.
Yeah.
That one will be a tougher one to sell, but yes, there's always a, yeah,
Over time.
yeah.
How many days after the treatment do you think you'll be able to conclusively, if you can conclusively destroy the tumor, how soon will you be able to discern that?
So usually the first cystoscopy after any kind of treatment is 3 months after. So we usually will wait for that time frame because the bladder needs time to heal. If you look inside after a treatment, everything is red and it's hard to tell what's going on. And that's the norm. And then we let everything heal, so we'll know at the 3-month mark, and that's usually a very telling sign.
At the 3 months
Yeah.
mark, and not before?
No. Most people will not look in at that point because you can't tell without, say, taking additional biopsies, for example.
Would the functioning of the bladder give you a clue to your success or failure?
It may. There are some drugs where we know the drug is working if patients are having side effects, right? So it may be that scenario. I don't know until we actually start treating the patients. So the patient who has no side effects, you may say, oh, did it actually do anything? But the patient who has some frequent urination for a period of time, that could be a signal that it was absorbed and the bladder is affected. It could simply also be a signal of adverse event. We don't know. Yeah.
So at that 3-month point, the patient comes back in, they get retested. Let's say it was not effective and the tumors recurred or come back. At that point, can you scrape again, or you can still take the bladder out at that point?
Well, we'd scrape again to see because you have to have definitive proof that there is recurrence and it's not simply a reaction. Because sometimes these tumors are just red, and it could simply be inflammation. And then theoretically, depending on how this evolves, you could probably retreat. Yeah.
So we are thinking right now of additional potential protocol where retreatment would be part of the protocol.
At that point, would you use the fluorescing of the cancer at all?
So this particular drug does not really fluoresce. The fluorescence is extremely weak. But what we have seen is that it has a very intense color. So wherever the drug is taken up by the tissue after washing, the urologist will see a very significant color change. Yeah. So we don't maybe even need to do fluorescence.
Okay.
But again, these are all based only on the animal experiments, right? We have to see what happens in the prime animal of all, humans.
All right, I guess that's everything then. Okay, thank you so much.
Big progress.
Yes, it's been excellent. Yeah.